Today we're going to talk about adaptive immunity versus innate immunity. Innate immunity is the non-specific response to any pathogen. And while adaptive immunity is more specific to one pathogen, and you gain that throughout your life. And at the first exposure of a pathogen, cells like macrophages, B cells, and dendritic cell will perform phagocytosis where they embraces the pathogen, bring it in using lysozyme, and then cut it into small fragments. And then your major histocompatibility complex will present that fragment to helper T cells. Major histocompatibility complex, there's two types of MHC. The first type is found in all nucleated body cells. That means the mature red blood cell is not included in this list. It displays peptide antigen to the cytotoxic T cell, while MHC2 is found in dendritic cells, macrophages, and B cells. After the MHC presented that foreign particle, the TCR, the receptor on helper T cell, would feel around this foreign particle and recognize it's not normal, and it will release a signaling molecule called the cytokine to both the B cell, which is produced in the bone marrow, and the cytotoxic T cell, which is produced in the thymus. The response for the B cells are humoral response, and the response produced by the T cell is called the cell-mediated response. So in adaptive immunity, there are two types of response, the humoral and the cell-mediated. The humoral is in the blood or lymph. The cell-mediated is where the specialized T cell destroy the infected host cell. And B cells, and there's a difference between the structure of B cells and T cells. From here, we can see that they're both a type of WBC, the white blood cell. They're called lymphocytes. And the B cell has that white shape, which is similar to the antibody. It's composed of a constant region, which is the same for every antibody region and any region on the B cell. But there's a variable region. It's specific to the antigen. And there's the receptor is composed of a heavy chain and a light chain, and they're connected by a disulfide bridge, which has to be broken by a strong reducer. And then when you look at the T cell, we have alpha chain and the beta chain that's also connected by a disulfide bridge. And there's also a variable region that's specific to the antigen, and the constant region that's the same for all the T cells. The variable region will only bind to the display presented by host cell, unlike the B cell that can recognize to the free-floating antigen. And the variation in that chain is caused by alternative splicing, as we discussed in mRNA. The helper T cell releases that signal to the B cell and the cytotoxic T cell. The function of the B cells are they disable the antigen with antibodies, and they're produced in bone marrow. While the function for the cytotoxic T cells are that they secrete proteins that trigger apoptosis of the infected cell only. The B cell will then go into the activated form, which is the plasma B cell or the memory B cell. The plasma B cell is in the blood, and it's short-lived, and they act fast. They produce the antibodies, which are immunoglobulins. They're just the B cell receptor that lack that transmembrane region. And they also produce the memory B cell, which are long-lived, and they produce the effector cells that will produce a short-lived and immediate response. Same thing with the cytotoxic T cell. It would have the active form and a memory form. The active form requires signal molecule from the helper T cell, and they would eliminate infected cell using apoptosis. And the memory cytotoxic T cell is the same thing with memory B cell. They're also long-lived. So the activated plasma B cell will produce the antibodies, the immunoglobulins, which are the B cell receptor without that transmembrane region. It's not burrowed in the B cell. It has three functions: neutralization, opsonization, activation of the complement system. In neutralization, the antibodies will attach to the pathogen, therefore disabling it from attaching to our own cell. And in opsonization, it would attach to the pathogen and signal that macrophage to perform phagocytosis, therefore destroying the pathogen. And in the third option, the activation of the complement system, it will signal the complement protein to form this complex that will recognize the antigen. and that membrane attacking complex forms a cylinder to punches through the pore to make a pore in the membrane that allows water and ions to enter, causing the cell to lyse. So all the plasma B cell and the memory B cells produce are the humoral response, which are the extracellular defense. Well, the memory cytotoxic T cell and the active cytotoxic T cell, they're all part of the cell-mediated response, which are intracellular defense. But now let's think about it. Our immune system is about recognition and memory. When we're exposed to the second time for the same pathogen, this will trigger our memory T cell, which are existing helper T cell that's not active the last time. And the secondary exposure will also trigger your memory B cell and your memory cytotoxic T cell that produce a faster response. So the memory T cell would also signal more memory B cell, while the memory T cell will signal more memory cytotoxic T cell.